Alright, in this lesson we are learning about enzymes. Here are our syllabus dot points. Now there's not a huge number of them, but they are quite specific, so please make sure you are following up on these by reading through them. Alright, let's start off. Enzymes are proteins, okay? They are globular proteins, so they are folded like that big ball, and they are biological catalysts, meaning that they speed up reactions without being altered themselves, right? They are made in cells and they are proteins. Enzymes can be found in all different places in the body, from the salivary glands where there's amylase to break down some sugars, all the way through the digestive system. What we are aiming to do when we are looking at an enzyme, is that we are taking a substrate and turning it into a product. So we are chemically changing one thing, which is the substrate, and turning it into something new, the product. These enzymes can be intracellular, so meaning they can be inside the cell, or they can work outside the cell in the bloodstream. Right, enzymes are really specific. You know that meme that says you had one job? That's pretty much them. They do not multitask. They can only catalyze one kind of reaction. That's it, right? That active site that they have is where the substrate binds. So, that's kind of the workbench. They are built and they are shaped to have a specific site available so the substrate can come and bind. And they often call it lock and key specificity, right? Only one key works in that lock, and that's what's happening. The substrate is the only thing that will fit into that active site or that workbench. So the shape and the chemical structure matches. Substrates, right, have to actually bind to that active site. Once the product is created, it can then leave. So once that's actually happened, it frees up the workbench. So really the enzyme is making all of that happen, but it's not actually being changed itself. There are 3 stages of catalysis, whatever we want to call it, and basically very simple. The substrate is binding to the active site, the substrate is being changed, and then the products that have been created are released and they vacate the active site. So the reaction may break down molecules like we're seeing here, or it might actually do the opposite, so it might actually be bonding them together. So nice little animation. You can see that a giant molecule is going to be broken down into 2 things there. We require that water molecule there to assist, and we're turning a disaccharide into 2 monosaccharides. Right, so there's lots of examples of enzymes here, and what you'll see in this particular section is you'll start to see that there is a bit of a pattern. If it ends in A's, it's generally an enzyme, okay? Lipase works on lipids, proteases work on proteins, so they start to name them based on what they do. Enzymes are proteins, so that means they can be denatured, remembering that denaturing is that irreversible structural change. Now, if the structure changes, the shape is going to change. That means the active site is going to change, and therefore the substrate can no longer bind, which means it had one job and it can no longer do it. And it has optimal conditions required for the optimal activity. So if you want an enzyme to be very efficient, you need to make sure it's in the optimum conditions. In order for things to actually hit the enzyme, so those substrates to get to the active site, we're really relying on random movements and collisions. So the substrates must actually collide with the enzyme in order for that reaction to take place. So the movement of the enzyme and the substrates are indeed random, but those collisions have to happen for the reaction to take place. Now, substrates are usually dissolved in water, and this both helps and hinders the process, right? It helps them because it gives them mobility to do that movement. However, there's so much space in a liquid for them to move around that, statistically speaking, there's not a lot of chance that they're going to run into an enzyme. So because of that difficulty aligning the substrate and the active site, many things impact the collisions occurring and therefore the overall activity of the enzyme and its ability to catalyze that reaction. The main things that are affecting enzyme activity: temperature, pH, and substrate concentration. Now, temperature affects the enzyme activity because of a few different reasons, right? So if you have an increase in heat energy, it means that the particles can start to get more of that kinetic energy and start moving a lot more, which actually increases the chance of collisions. However, if that goes too far and we have too much heat energy, we actually start to change the bonds in that protein, and therefore it can denature. So there's that tiny little realm, so you might call it the Goldilocks region, where it's working really well, but then as soon as we get too hot and it starts to denature, that activity, so the enzyme activity, is going to drop massively. Now, pH pH affects enzyme activity in lots of different ways. Now, it depends where the enzyme is working as to what it's adapted to do. So digestive enzymes in the stomach, for example, might work at really low pHs because it's very acidic in the stomach, but a blood, or in this case, say, salivary amylase might work at a more neutral pH. It really does depend where you're finding them in the body. Any change away from that optimum pH will decrease activity completely. So we can see here pepsin, which is found in the stomach, so really acidic environment where it's working the highest. The salivary amylase is in your mouth, so again around that neutral environment. And arginase is found in the liver, and it's actually working ideally at a more basic level there. And substrate concentration affects enzyme activity. Okay, if there's decreased concentration, then there's decreased chance of the collision. So remembering those substrate molecules have to collide with the enzyme active site to be catalyzed. If you increase the substrate, you actually start to occupy all those active sites. So what's going to happen is there's going to be a max out point, and you can see that here. Once it reaches maximum activity, it's not going to get any higher, and that's all that's going to happen in that enzyme reaction. Enzyme activity can be used productively, and mostly they are talking about immobilized enzymes. Now, immobilized enzymes are those that are in fact attached to another material to restrict their movement, and they're frequently used in the food industry and the medicinal industry and things like that. Okay, now there's advantages to immobilizing those enzymes. If you've stuck them somewhere, you're in control of that reaction a lot more. You can stop it or start it on command, right? As opposed to just putting them in solution and hoping they work when you want them to work. You can recycle the enzymes, okay? You can reuse them as opposed to just putting them in solution and not being able to retrieve them out. So you can use them multiple times. You have increased enzyme stability with condition changes. It means if they're not, in the right pH or the right temperature, you can degrade those enzymes much faster. Therefore, you can't reuse them. So you want to be able to make sure, well, actually, that's not the right temperature. We'll take them out, fix the conditions, and then put them back in. And you can actually use a higher concentration of enzymes because you're not just putting them in solution and hoping that you haven't saturated that solution. The random collisions aren't really that very efficient, so you can increase that surface area of enzymes and then control the concentration. And lactose-free milk is one of the products that we make using immobilized enzymes. So lactose-free milk, you can use it if you've got an allergy or an intolerance to lactose, which is the sugar found in milk. And lactase, so there's that A's on the end again, is the part of the enzyme that's going to do that breakdown. And it's breaking down lactose, which is a disaccharide, into glucose and galactose. So it's a hydrolysis reaction. So some people who are intolerant don't actually have lactase in their system, or it doesn't work. Now, we'll go back. Some of the benefits of that is these two sugars are actually a lot sweeter on their own than lactase, so you don't have to add any added sugar to some products. The texture, it makes a different kind of ice cream texture when it freezes, and also glucose and galactose can be fermented quicker, so you can make better cheeses and yogurts. So there are our syllabus dot points. There's quite a bit in this particular guidance and application section, but making sure you understand the basics of the enzymes there.